Hello everyone. So welcome to today's class. Today we'll talk about GAT-B 2020 last 5 years solved questions, and today's topic is biotechniques. So let me see who all are online. Okay. So I have to see who all are online and then we can start. I'll wait for people to join and we'll start the live session. So let me put it in the group such that people can get a notification that I'm online. So say hi if you are online. Yeah. So who all are online, guys? Tell me please. I can see 4 people are online. Okay. So Vagdevi, Tamagna. So hi everyone. So today's topic is question solving from GAT-B 2020, I mean JNU CEB papers, because previously it was known as JNU CEB exam. Now it is known as GAT-B 2020. So you just need to know the type of questions, what are the depth of the question, what are the topics that are asked, how can you prepare, right? So do you guys know what are the resources that you can use to prepare this section? कौन सा वाला resource आप लोग use कर सकते हो, क्या बेहतर होगा, right, आपके लिए? तो चलिए थोड़ा देर वेट कर जाते हैं, लोगों को ज्वाइन करने दीजिए एंड देन आई स्टार्ट. ओके, सो प्लीज से हाई जब जो लोग ऑनलाइन है एंड लेट मी नो योर नेम्स. सो टुडेज गोल इज we'll do 25 questions in 25 minutes. Is it possible? So let's see, we'll make it possible. Okay, so you have to work very fast and hit your answer in the comments. Okay, so you have to give your answer in the comments. So everybody is ready? Everybody is with me? Everybody is with me, guys? So shall I start? Shall I start, guys? Okay, so here is a question from JNU CEB 2018. We are talking about biological techniques, or the techniques that are used in biology. So these are very important concepts, low hanging fruit, because it can give you quite a lot of marks. So it's good. So guys, tell me this answer. Secondary structure content of a protein cannot be determined by which of the following techniques? Series spectroscopy, NMR spectroscopy, X-ray crystallography, Foster resonance energy transfer based fluorescence spectroscopy. So can you guys tell me which one of these would be the proper answer? Type your answer in the comment section. Come on. This is the first question. Come on guys, waiting for your answer, waiting for your answer. You cannot take more than 1 minute for these kind of questions. ठीक है, 1 मिनट से ज़्यादा नहीं ले सकते. Okay, Saurav Das is saying answer 4. Anybody else? So you have to type your answer, means like you say A, B or C, D, something like that. Okay. So Rupa Chatterjee is saying D. So Rupa, you are also a new person in the this series. So this is a series, okay? GAT-B question. So at least there are 4 or 5 videos previously done. So you have to check in Unacademy-JAM live page in this YouTube channel of Unacademy. So such that you can get the benefit out of it. Okay. So everybody is saying D and the correct answer is D. So let me tell you guys, this is a very popular type of question. So CD spectroscopy is exclusively give you idea about, I mean it can give you idea nicely about secondary structures. Okay, but not tertiary structures or higher order structures. Now, NMR and X-ray crystallography can give you information about primary, secondary, and tertiary. All of these structures it can give information about. So guys, if you wonder that where I can get these kind of materials or where I can get these kind of exam point of view from this particular topic, I should tell you that I have a crash course on that, okay? So I have a crash course on that in Unacademy Plus platform, and this is very cheap. It's only 1800 rupees, can be doable within a month. So I have a crash course for GAT-B 2020, so which is doable in a month. In a month time you can do it, so you can invest that. It has more than 300 lectures. Okay, so moving on to the next point. The rate of migration of DNA fragments through an agarose gel is determined by number of nucleotides in the fragment, ratio of adenine to cytosine, hydrogen bonds between the base pairs, volume of the sample. So can you guys tell me, have you guys done these kind of experiments like agarose gel electrophoresis in your college? Saurav Das is saying that the answer would be A. So Tamagna Chakraborty is also saying the answer would be A. So a lot of bongs are here today. So let me get the answer from everybody and then I'll say. So the questions are arranged in a way that we are going to start with the easier ones and then we would eventually ramp up to the difficult ones, okay? So anybody else? What is their answer? I wanted to know. Come on, don't be shy, give your answers. Option A would be correct. But somebody said, such as Bhagdevi said, it would be B. So the ratio of adenine or cytosine has nothing to do with the electrophoretic mobility. So the electrophoretic mobility, so why does DNA move in a gel under an electric field? So if this is a gel, so I'll tell you, if this is a gel which is very deformed and bad because I have casted this gel. So anyway, so here you would have the molecular ladders and let's say here would be your band, okay? So this band would correspond to a DNA which has a smaller size, whereas this band would correspond to a DNA which has a bigger size. And DNA migrates because it has a negative charge due to the phosphate backbone, so it would migrate from negative to positive electrodes, right? That we know. So it's a function of charge, size, etc., but not a function of what type of base is inside that DNA. So the constituent does not like affect the mobility, okay? So everybody understand this part? Again, I'm saying these kind of concepts which are especially important for exam are shown in these crash course, which would be very, very beneficial, and it would be only 1800 rupees for you guys. And definitely you can use my code AP10. I mean, don't forget to use this, otherwise you won't get this price. So you can use AP10 to avail this thing. So you just need to go to the Plus app, Unacademy, and you have to get this course. Okay, so the undifferentiated and unorganized mass of cell in the tissue culture is known as plant tissue culture. It should be written here, the plant, which was not written here, but what can we do? So it's in plant tissue culture. What is that? So tell me guys, what would be the answer? Okay, I mean, this was a bit of a mistake for me because I thought that I would show you a callus culture, what does callus culture look like, and I put it without animating it. So anyway, everybody got it. So yeah, moving on to the next one. In recombinant DNA technology, a DNA probe is used to detect specific nucleotide sequence, insert a foreign gene, digest a genomic DNA, clone extra piece of DNA. So guys, type your answer. This is fairly easy. So Tamagna is saying A. What about L? What about others? সই করে নিয়ে নাও। সই করেছো? সই তো করতে হবে না? হ্যাঁ? Guys, I have to go for a while, just a little bit, I'm so sorry about it, but I have to go for a while, just 2 minutes. Hold on there, I'll come back. Okay, I'll come back, give me a little bit time. Okay guys, I'm back. So the question is one, it's fairly easy, right? So a DNA probe is used to detect a complementary DNA sequence or RNA sequence, doesn't matter. So these kind of things are used in hybridization techniques such as southern blot, western blot, I mean not western blot, but southern blot, northern blot, etc., right? So in order to insert a gene, what set of enzymes do you use? You use restriction enzymes, right? Restriction endonuclease. Then, I mean, also, which you don't know, let me tell you, you could also use recombinase. So there are cloning methods which do not use restriction endonuclease or ligase. You would be amused, like, okay, what is happening? We knew always cloning requires these enzymes, but you might not need all of these enzymes if you use recombinase based cloning system. This recombinase based cloning system is dependent upon site directed mutagenesis. Okay, and this is very important. This is the way by which a phage gets inside in the host E. coli genome. Okay, digestion of the DNA, obviously you digest it with either endonuclease / exonuclease as well, and cloning an extra piece of DNA, right? Cloning, right? So you can PCR amplify, you can put it in the bacteria, you can amplify all of these things. So everybody, this part is clear, right? Now look at this big ass question. So this question says a researcher did the following step of the experiment: isolated the genomic DNA from a microorganism and purified it fine, digested the DNA sample with HindIII restriction enzyme overnight, performed the agarose gel electrophoresis of digested sample. After electrophoresis, transferred the separated band to the nitrocellulose membrane, prepared the gene-based radioactive probe and hybridized with nitrocellulose membrane, checked it in an autoradiograph. So obviously they have depicted a experimental technique. You just have to guess what is this experimental technique. Okay? So obviously this was very easy, it's just a southern blot. One other type of question that can be asked from these kind of concepts is basically they would give you some random haphazard sequence of these and you have to arrange or find the correct order. Okay, so you have to say A, then B, then C or something like that, or A, then C, then D, then E, something like that. Okay, so you have to arrange that. So maybe they would jumble these 1, 2, 3, 4 and you have to get the correct sequence of it. This is another flavor where question can be asked. So about many of these questions we have collected and made a text booklet. So do let me know if you need a test booklet like that from a biotechnics, because it is not available in the question books so much nicely. So I have a booklet on that. You can email me for that. Okay, I'm giving you my email address. सबको मजा आ रहा है ना? So this is my email address. You can email if you need a booklet like this, which would be helping you to prepare. And obviously, the crash course in GAT-B 2020 has it all. So there is a dedicated 10-day lecture series for these biology techniques where we take every day, we take a theme, like for example, working with DNA, working with RNA, working with proteins, biophysical techniques. We take a theme each day and discuss the techniques, their principles, what type of question can be asked in that course, okay? If you are interested, it's only 1800 rupees, 1800 rupees, kapde ka daam mein course mil raha hai yaar. Chalo, then they said a protein solution after 100-fold dilution have an absorbance of 0.04 in 1 centimeter path length of a cuvette. And they have given a absorbance wavelength, the light that is used. The molar extinction coefficient is 44000 mole inverse, centimeter inverse. Find out the approximate concentration of the stock solution. So guys, take a pen and paper, try to solve this question and try to give the answer. So I'll do the solving with you guys as well. Okay, so try to solve it by yourself and give me the answer. Extinction coefficient equal to 44, right? Then you have path length, which is L, equal to 1 centimeter, right? So these are the information that you are provided with, okay? And you know Beer-Lambert law, absorbance is proportional to concentration. So absorbance is, and also absorbance is proportional to the path length. So about Beer-Lambert law, about derivation of Beer-Lambert law, I have detailed lecture in this particular course. But for now, I'm just solving it quickly, right? So this is the formula that you have to use, right? And remember one thing, this was 1000-fold dilution. So whatever you would get the absorbance of, this is the diluted sample, right? So basically you have to multiply with 10 to the power 3 to get the proper concentration, right guys? Right guys? So come on, try to answer this question. So let me also solve in my way, you also solve in your way, okay? So absorbance equal to ECL. So obviously concentration would be absorbance by concentration by L. So let me plug in the value. So this is 0.4, then it would be 44 into, so let me just get rid of this and put a 10 to the power 4 here, okay? So I can do that, right? Simple mathematics. Yeah, so let me get back. Okay, so and path length is 1 centimeter, right? And remember the unit, guys, remember the unit. So the unit for the molar extinction coefficient was mole inverse 1, centimeter inverse 1. Here the path length was centimeter, so basically this centimeter goes off. So whatever your unit would be ultimately is actually in molars, okay? So basically you have 1 divided by 11 into 10 to the power minus 4, right? 10 to the power minus 4. So this is the concentration of your diluted stock, right? And your concentrated stock need to be multiplied with 10 to the power 3. So if we multiply that, you would have 1 by 11 into 10 to the power minus 1, right? Okay guys, and the number comes something around like this, 0.009, okay, 0.00909. So I'm putting it in the calculator, okay, this much molar, right, this much molar. Now the answers, look at it, everything is in millimolar. So obviously whatever it is in molar, you cannot see the particular thing, so this one would not be the answer, any of these three would be the answer. So in order to do millimolar, what we would do, so we have to shift the point 3 times, right? So we have to shift the point like this, right, 10 to the power minus 3 molar, which is also written as 9 into millimolar, right? So this is the answer, right? That means it's like almost equal to 9.1. So 9.1 millimolar is the answer, right guys? Got it? Yep. So we got the answer. So these kind of little bit of mathematical question can be asked. So which of the following method will you use to determine, differentiate between species and a strain? So try to answer this question, guys. Try to answer this question. So, which of the following methods you would be using? DNA footprinting, whole cell protein profiling like the proteome, the DNA fingerprinting, etc. So obviously Saurav is correct. I think Saurav has taken a booklet and which helped him a lot, right Saurav? You're the same Saurav in the plus class, right? This is a very common name, that's why I am asking. So I hope the booklet that I provided was very helpful. People get a lot of idea about these biotechniques and all. Yeah, so A is the correct answer. So Raju Kumar, Saurav, both of you guys are right. So what you can see that these questions are not so deep. Little bit of understanding of these techniques, the principle of these techniques, their application is more than enough to solve these questions. All you need to do is practice more and more questions from this, read the basics, and that's all. The basics you can always get from my crash course. The practice question you can also get from the crash course, or you can get the booklet from me. You can email me for the booklet. Okay guys, let me tell you something. If you really want to get a plus subscription, which might be useful for you, so you can email me, you can give me the email, you can email me, I can tell you, I can send you the booklet, okay? Okay guys? So let me tell you something about the IIT-JAM subscription. So if you have 1 month subscription, it's 2000 rupees, but with my code AP10, if you use this filter, you would have 1800 rupees per month. Perfect for a GAT-B preparation. GAT-B 2020 crash course, okay? If you are preparing for IIT-JAM next year, this would be very useful, this or this, depending upon you're in first year or in third year, this would be, or second year, this would be best. At least guys, taking a 3-month course would be better than 1 month. You can also try for 1 month and then extend it, so it's your decision, but this is very cheap compared to any other education center or education institute, and the quality, I can give you assurance of that. Okay, since Saurav is the plus student, why don't you talk about a little bit about the quality of the classes, right? A little bit about the classes, like I provide 24 classes in a month, so obviously cost per class is 46 if you take a 12 month subscription. Otherwise, the cost is so low that it would be lower than your fast food budget. And obviously, you have a live interface where you can solve questions, you can answer in the polls, and definitely you can get a feeling of these kind of question answering or solving. So it would be really good. Obviously, there is free classes in the learning app. You can download the learning app, avail all the free classes, but there would be paid classes. The difference between these two, guys, the difference is the paid classes, they are structured. So obviously, this is the web series, and this is basically the trailer. Okay? So obviously, this is overall the idea about it. So the paid classes also gives you a flavor of question solving, so your confidence in the exam hall, or you get more familiarized with the online interface of answering, right? So which would help you in the exam day. You won't even notice that, but it would really, oh, my email you need it? Okay, I'll give you my email. Sorry. Okay. You can also send me your WhatsApp number in my email. We can chat in WhatsApp as well. I'm okay with anything. Okay, guys. Also in Unacademy, which I provide, is quite a lot of these kind of booklets and these kind of like flashcards, which would help you to prepare for this thing, right? So the live interaction is best in the Unacademy Plus platform. At least the students are saying that. So we really interact a lot. So let's move on. So I give you a pretty much idea about what happens in the Plus platform. So you can really increase the family. There are quite a lot of students there, and it's really fun to interact with them. So definitely we move on to the next question. So let's try to answer this. An absorption spectrum of a solution is given below. Choose the correct statement regarding the solution. Okay, the solution contains only DNA. So this is the UV vis spectroscopy reading spectrum, UV vis spectrum. Okay, so you get some peak, and you have to understand these peak correspond to what. Okay, okay, guys, tell me. Tell me, which would be the answer? Which would be the answer, guys? Answer would be C. Saurav is saying answer would be C. Shubhajit Giri is saying the answer would be C, and the answer is C. So first of all, 260. This is for DNA or RNA, right? 280 for aromatic amino acids in the protein, right? Such as tryptophan, phenylalanine, etc., right? So all these aromatic amino acids give a band in 280. Now 480, it might be due to a prosthetic group, but 480 nanometer wavelength is for a blue color. So obviously 480 is the illumination for GFP as well. So you illuminate in blue color, you get a fluorescence in green color. So this is important, okay? Right? So this question was very simple and dependent upon the principle of UV vis spectrophotometry, and it was very easy, right? So you have to know people use A260 by A280 ratio to calculate the purity of DNA. So for DNA, this ideal ratio should be 1.8. For RNA, this ratio should be 2, right? For example, if they say in an RNA sample that is extracted by you, that A260 divided by A280 is actually coming to 1.6. That implies what type of contamination in the sample? So what do you see? What type of contamination you can say in the sample? The answers are protein, DNA, carbohydrate, and 4 is ketone bodies. So what would be the appropriate answer, guys? Protein contamination, right? No, 280, who is absorbing? Protein, right? At A280, who is absorbing? Protein, right? So if you have this thing, so imagine if you have a value for A280, then let's say so in ideal cases, protein is very low. I mean, never you get a pure sample, totally pure sample. Let's say your protein value is 2 and your A260 by 280 is something, these numbers are arbitrary, right? 10. So your overall would be 5, right? But here, imagine, here what would happen? If you have too much protein, then it would be 5, so it would be 2, right? So whenever your A260 280 drops, A260 280 drops, that means you have protein contamination. Understood, guys? Now let's try to look at the time. Okay, it's almost 30 minutes, so I have to finish the class, but anyway we'll try to practice as much as possible. So the resolving power of the microscope can be increased by which of the following? Read these things: increasing the diameter of the eyepiece, decreasing the diameter of the eyepiece, increasing the diameter of the objective lens, decreasing the diameter of the objective. So just to let you know, I have a dedicated class for microscopy, all the modern microscopy technique with live example. Nobody would give you that how microscopes are assembled and all of these things. So those things are shown in the plus course. Okay, how microscopes are assembled, all those images are there. How the light, so I showed you a naked microscope, like not a microscope image downloaded from a Google thing. So I have shown you in this crash course how microscopes are assembled, how laser line move, etc. Okay, so that is why this course could be very attractive for you guys. So there is a course which is known as Biotechnics Quick Revision, which includes all of these things. So now give me the answer. Which would be the answer, guys? Which would be the answer? So I'll be waiting for your answer. So I hope this session was nice. Probably this would be our last question answer. We could not finish all of that, but maza aaya, right? We have looked at quite a lot of questions and we tried to solve it together. And most of you have done very nicely, but a little bit of the cases you might have answered not properly or might not have answered. So in that cases, you need to practice more. In order to get the booklet, you can definitely email me. Again, my email is provided here. Here Raju is saying answer B. So Raju's answer is B. Anybody is giving any other answer? So when these kind of questions are asked, understand the logic. Be very sure from your end. If you're sure, then answer, otherwise don't answer. Playing this game is very important. And what you need to do is you need to play the game. Don't get negative. That's the game. Okay, guys? So tell me, which would be the answer? Anybody else? So Rupa Chatterjee is saying it would be B, decreasing the diameter of eyepiece. So now, in order to solve this question, let's solve this question. So we have to understand on what factor resolving power of the microscope depends on. So we don't have a detailed video on that right now. I mean, we don't have a time for a detailed explanation for that. But let me tell you, resolving power, I hope my handwriting is understandable, is actually 2 mu sine theta. I'll explain what these are, lambda, okay? Wavelength of the light used. What is sine theta? I'll come to it. Mu is refractive index of the media. And what is sine theta? So if we try to draw an objective lens, so this is the objective lens, okay, which is badly deformed in my hand. So basically this is the part of the objective, okay, this is the objective you are seeing, okay, and this is your specimen plane, okay, this is your specimen plane, your glass slide, and maybe your specimen is here. For a what should I say, for clarity's sake, this specimen is a transparent specimen, so it's a point specimen, okay. Now obviously this is your optical axis, which is theta, should not be theta, but it is theta, and this is the cone of light that should be collected by the objective, right. So basically look at this triangle, okay, this triangle, and what is theta? This half angle with the optical axis is theta, right. So imagine, and what is the diameter of the objective? This is the diameter of the objective, right, objective lens. So imagine you have shrunken the diameter of the objective, right. This was the objective, and now your diameter is low. So what would happen, guys? Where do you find the change? You would find the change in theta, right. So theta new would be decreasing than theta old, right. So when you decrease the diameter, right, when you decrease the diameter, your theta would be decreased, right, and resolving power is directly proportional to theta, right, sine theta, right. When theta is low, sine theta would also be low, right, guys, sine theta's value would be low. As a result, what would happen? If you decrease theta, what would happen, guys? If you decrease theta, your resolving power would decrease. If you increase theta, resolving power would increase, right. So definitely it happens with the objective lens. So you have to increase the diameter of the objective. So by increasing theta, you increase this thing. So by increasing the diameter of the lens, what happens, guys? More light gets in. And according to Ernst Abbe, the quality of image depends upon the number of diffraction order that you are collecting. Greater the diffraction order with the image quality. About diffraction order, about image quality, about Abbe's diffraction limit, all of these things are depicted in this course, which cost only 1800 rupees only. So if you guys are interested, to subscribe to the Plus platform, the learning app, and the best part is you can learn whenever you want, wherever you want. You are doing yoga and you want to learn and listen to my lectures, you can get motivation and do the yoga at the same time. So that's a good part. So everybody, I'll say goodbye to you guys. I hope this session was very useful for you guys. Do let me know in the comment whether this session was useful or not. Give me some feedback. You need to give me some feedback in the comment, otherwise how would I know? and I'll do more such session. Tomorrow there is another session, okay? So do join the session. The session is in the same time, okay? And if you need the booklets, do let me know. I'm putting my email once again. Okay, bye everyone. I'm putting my email again. If anybody want to get the booklet, I'll tell you. Okay, chalo, bye.